Thank you much. Thanks much. Thank you also for the invitation, guys. It was a pleasure. So before I get into the specifics of what this whole thing is going to be, I want to go back to something Nakum said. So he talked about internal representations, and I guess in some sense, and someone had a question too about whether an insect is a robot, or someone asked that somewhere there. And I guess this is something I think about a fair bit. So this is everyone's popular conception of an insect, maybe this cartoon, the stimulus response, it's sort of a reflex machine. As it turns out, reflexes are actually cooler and more interesting than you might think, as people like Gwyneth Card and others have shown. There's a lot of complexity there. But by the end of the talk, you can tell me if you think there's maybe more going on than just reflexes stuff, because in my opinion, there is, even in the little fly, okay? So like Nakum said, navigation provides a good way to explore internal representations, and that's sort of our angle as well. In our case, we do not work on the fantastic, vast range of beautiful animals, some of which you've already heard about from Marie, from Stanley just now. Stanley's worked on this creature, the monarch butterfly. He's worked on bees. I mean, maybe not this bee, but maybe he has. Actually, I don't know. Maybe he's worked on that too. Oh, yes, he has. There's bumblebee. You worked on the bumblebee too. So, yeah. So, and there's people here from Rudiger's lab. So, obviously, all of you know that these animals have amazing navigational abilities. They can go off to forage, come back home, tell their buddies about it, and then the buddies go off and find food as well, like the honeybee. So these are great navigators. An animal that you don't typically associate with tremendous feats of navigation is perhaps this one. Of course, our interest is entirely because of its inner beauty, and I refer to, it really is quite amazing. So I'm going to start by just telling you a little bit about, this is sort of the reductionist view of beauty, right? Nakum was, kind of saying, well, reductionism has its role, but really, come on. And so this will be kind of the counter a little bit. I mean, I absolutely think it's amazing to work in all these lovely systems, absolutely amazing. But the fly has its advantages. And so perhaps the biggest, the most powerful is actually something coming from yeast. So it's not even a fly thing. It's the yeast transcription system that is used in the fly. And it's just been revolutionary. This is not the only system. There's multiples of these transcription systems. But what they allow you to do is essentially separate things about where you want to express a particular protein and which protein you want expressed. Okay? So what does that actually mean? So the bottom line for this particular transcription system is that you have something called an upstream activator sequence. Every time this is activated, you're going to get the production of some protein. Okay? So in this case, let's say it's GCAMP, a calcium indicator that in this case is made at Janelia that we use a lot. And so, but you control the triggering of this upstream activator sequence is through something called GAL4. Okay? This GAL4, however, is produced by, let's say, a different line of flies that you have to cross into this line of flies in order to actually get the whole thing to work. So imagine a lock and key system. You have something that triggers the production of GAL4, which might be some cell-specific promoter that tells you, just express something just in the eyes or just in the limbs or in the muscles or whatever else. So that would produce the GAL4, but in a fly that just had this bit, nothing else would happen. Okay? There's no production of any other proteins like GCAMP or anything else. You have another set of flies where it's UAS and let's say GCAMP, but unless this GAL4 is present, nothing happens. The magic is you cross this, the progeny now have both of these, and so you can get GAL4 and UAS and get your particular indicator, maybe in this case, specifically in the neurons of interest. Okay? So you can maintain, and this is what in fact we do, a ton of different GAL4 lines. And so, for example, there's thousands and thousands that are available to the community. There's a catalog, and you can literally go in and say, give me this particular GAL4 line, and this will allow me to maybe target neurons specifically in one particular brain region, and even there within a particular type. So think of Cree locks, but really on steroids. Okay, so you have thousands of these, and you have many, many different UAS lines. So you can have something like a channel rhodopsin, you can have GFP, GCAMP, whatever else. And this combination is kind of the key behind a lot of fly experiments. So the kind of specificity we get, I mean, Stanley's already talked to you about the wonders of the insect brain. You have identified neurons, so you can come back and record from the same ones time after time after time. But the advantage here is that they'd be colored for you. So you can make some of them green, some of them red, et cetera, and you can label just these neurons and no other neurons in the entire brain. So there's over 100,000 neurons here, but just these guys labeled, right? So, yeah.
I'm just curious if, I just noticed that you're using enhancer-driven instead of promoter. Is that how you get such?
Yeah, you can get, I mean, so actually, as it turns out, those enhancer trap lines, the original enhancer trap lines, now you go to little tiny fragments of those. So you get increasingly specific, little elements that allow you to target very small numbers of cells, but yes. And actually, I should mention also that in some sense, we're almost not looking at which gene we're even talking about. That's a guide to maybe isolate some particular set of neurons, but that's it. After that, it's just, you can do it almost randomly and come up with really interesting combinations. So basically, you have an addressing system that allows you to put a protein of interest in just a small number of cells. And this is what we'll use again and again. But of course, the whole point of this thing I claimed was that we wanted to study internal representations and specifically using navigation. And so the key question is, can flies even navigate? Or in what ways can they navigate? And so there's a lot of different lines of evidence suggesting that they can navigate. And so one recent article that I like quite a bit came from Michael Dickinson's lab, and that shows, so the article was titled, had to do with path integration, but the form in which they measure it here is that they place a single fly in a little arena, it's not very large, and there's a central spot that becomes significant after food, but is previously not significant, okay? So initially, before the food arrives, the fly sort of goes every which way. But then once it's just tasted the food once, so after that the food is removed, so there's a little slide and they slide out the food, so there's now no food there. The fly tends to come back, circle around, come back. So it's as if once just having known that the food is in a particular place, this is now done in darkness, the fly will just go in these little loops and it'll keep coming back. And so when they analyzed data not just from one fly but from all flies, and they saw that it was clustered around this middle thing, when they analyzed the trajectories, they found something really cool. So what Irene Kim found was that depending on how far away the fly went from that central spot where the food was, the turns the fly would take would kind of reflect the fact that it seemed to know how far away it actually was. So if it was close by, it would take these turns, take another turn, come back to circle around. If it was further away, it would take a longer time to make that first turn and then come back. So the little analysis things they did told them that it was likely that the fly was storing things like distances and angles somewhere, and maybe all the way to a full-fledged path integration was going on. So that's a fly in a very local sense using path integration, right? This is not like over hundreds of meters or something. But the fact that it can do that for its body size, it's actually quite significant. And then there's additional evidence that you heard from Nakhum about, and you'll hear perhaps even more tomorrow and so on, about the Morris water maze. And so Michael Reiser actually did a version of the Morris water maze. So in the Morris water maze, there's this large tank, and then it's not this large, it's actually much larger. And then you have an immersed little platform there. It's milky white water, rodent introduced there, and eventually, usually with the experimenter putting the thing eventually on the platform, but sometimes it figures it out on its own and kind of realizes there's a platform there, so that the next time you put it, it gets a bit better at finding its way to the platform, so on and so forth, doesn't like being in open water. So the idea was to push that into the fly. And so if you actually use a water-based strategy like that for the fly, it's a very short experiment. So instead, what Michael decided to do was actually use a thermal maze. So here what he's doing is it's about this large. He's got one spot in there that with the Peltier element he makes cool at 25 degrees. And he actually, in this case, is Tyler Ofstad, who's a graduate student with Charles Zucker and Michael Reiser. And everything else is hot. It's 35 degrees, okay? And around this is a visual arena, an LED display to give you kind of a visual surrounding. And the experiment is going to be quite simple. All he's going to do is every 3 minutes or so, he's going to take this cool spot and bump it to a different place. The fly doesn't like being in the warm spot. It likes to be in the cooler area. The question is, if he moves it to a different spot, can the flies use the fact that the visual cues, which also move with the spot, so it's coupled, can they use that to find the spot faster and faster? Okay? So this would be the equivalent of taking the rodent out of the pool and putting it back in, and all the way to the probe trial, where there is no thermal cue. It's just, can they find it still based purely on the visual surroundings? So this is published work. I'm just going to show you one movie, which I've shown 1,000,000 times, and it's still something I can watch again. So you're going to have to suffer through it if you've seen it 1,000,000 times. Some of you have. So in this movie, where the flies are each individually tracked, this is software from Kristen Branson, each of these is a little fly, and they're all tracked. And this little dotted line is going to be where that cool spot is, okay? And the flies obviously can't see the dotted line, it's just for you. And the spot is going to move around between different positions, and every time the spot moves, this whole world will rotate with the spot, okay? So it's the coupled condition. And what they were measuring is how long does it take the flies to find this target, which is what is plotted on the y-axis as the number of trials go up. I'm just going to run you through a few trials. This is sped up, so they're not actually running on quite as fast as you see here, but it's not sped up so much that you don't get a good idea for what they're doing. Initially, they sort of pause there and then keep going. It's after a while that they seem to realize, okay, if I find it, I need to stick there because it's really not a good idea. So then they stick. Then it jumps, but the whole world moves. Some of them are now, you'll notice no one's going in and going out. They go there, they stick, right? So they've already changed their strategy. Most of them have. So now, there we go again. You can already see that the time, well, okay, again, right? I mean, some of you probably would get lost if you were kind of going out on the slopes as well. I did anyway. So it's going to fluctuate a little bit in terms of the time to target, but eventually it's going to settle to something quite low. So there are some star performers who, when the spot moves, they'll just go straight to it now, okay? And so again, it'll fluctuate and eventually ends up down there by the end. I'm just going to play it because I'm sure some of you are still enjoying watching it. And so there's many experiments and questions you could ask about, is there a social effect or so on and so forth. They did these experiments in single flies, and they could still do this task so quite well. Anyway, so you get the idea. The cool thing for us is that the region of the brain that was implicated in their experiments, so they could do these experiments, behavioral genetics experiments, where they could silence selectively using, again, these GAL4 lines, just particular types of neurons and ask which neurons were important for which aspect of the task. So you don't want to silence a bunch of neurons and find out, okay, the fly can't walk, and so of course it can't do the task. You want them to be able to walk. You want them to be able to sense thermal cues, but maybe not make the association between the visual landmark and the specific spot. And so when you pin it down to that particular visual kind of motor thing, the region that popped up in these particular kinds of tasks was the central complex. That's also the region that's come up in Roland Strauss's experiments in a sort of working memory-like task for landmarks. So this is a task where there's a bright kind of background and a dark stripe on one side, another dark stripe on the other, and the fly will kind of pace its cage endlessly back and forth. But then you take away those things and ask, will the fly still maintain a sense of where it needs to go? And it does. And you can even make it so that you throw on a distractor stripe, say somewhere off to the side, have the fly go there, and then take away everything and ask, does it remember to come back to where the original stripes used to be? And they do. And once again, the key to get this working memory, the short-term memory, working memory is a strong term, so let's just say short-term orientation memory, the key to that turns out to be, again, the central complex. And then finally, I'll mention work from Reinhard Wolf and Martin Heisenberg and their colleagues, Li Liu, Aike Guo, showing that for visual pattern memory, so this is memories where you have a fly in a little virtual reality simulator thing, so it's flapping its wings, and it's on a torque motor, it's sort of tethered, it can move around, and every time it sees one part of the visual scene, it's hit with a laser on its butt. It doesn't like that very much, and so it learns over time to fixate on other parts of the visual scene. And again, this is the kind of thing that, there's evidence suggesting the central complex is involved in that as well. So a lot of this has been behavioral genetics types of evidence. But all of it has suggested, something for the central complex. Now, the dirty little secret about these sorts of experiments in general is that, of course, when you have these GAL4 drivers, you tend to go where the drivers point you. So if you have a line that labels something that looks extremely interesting and exciting, you tend to test those perhaps more often than something that labels random neuropil. So there's a bit of a bias involved in just which GAL4 lines are available that you should know. Since this is a winter school, this is something you should always ask, like how broad are the lines they tested? Is it really sampling the whole of the brain and this is the only region, or is it, how specific is this stuff really? But in this particular case, at least in some of these cases, it really does point to at least the central role for the central complex, okay? So just quickly about the central complex, Stanley's given you a fantastic introduction to it in all kinds of insects. This is going to be slightly more Drosophila specific. So there's a few thousand neurons in the fly in the central complex, and they connect these different structures. As Stanley's already explained to you, these structures are really neuropil structures. These are not cell bodies there. The cell bodies are off somewhere to the side, and they send projections, and the arbors define these structures. The interactions between these different cell types define them. And there's layers and columns, as you saw in Stanley's talk already. There's over 100 cell types. So this is the nasty thing about the central complex from the perspective of complexity. I mean, there's a lot of cell types here. It's not so trivial to kind of nail down exactly what's going on in this region. And we, as Stanley again hinted, there's tons of stuff we think the central complex is doing. It's not just doing, you're going to hear something about a ring attractor, but it's really much more than that, okay? So this was all behavior. On the physiological side, back when we started, almost all the work had come from other systems. So this is Uwe Homburg's lab's work. There's multiple people here who have links to Uwe either through Stanley or directly or through Keram Pfeiffer, or he has many progeny who are exploring many different insects. But they've done beautiful work showing the impact of polarized light on responses in the central complex. And then you've heard, I'm sure, from Marie. Basel in her lab did electrophysiology in the dung beetle. Again, like here, there's a beautiful connection to the behavior that they've highlighted as well. The monarch butterfly, Stanley did work when he was with Stanley Reppert on just like fantastic recordings suggesting that there are similarities between the types of neurons across these systems. And then there's extracellular recordings from Roy Ritzman's lab in the cockroach. These were from unidentified neurons, so I should mention, this is key. These recordings, they were in immobilized animals where the neurons were known, so they knew which neurons they were recording from every time, and that turns out to be really important in the central complex. But the magic of the cockroach is it's this tank-like animal, and these guys have succeeded in doing extracellular recordings from animals that are moving around. And one of the things they've shown that's really cool is that if you stimulate these neurons, not just record from them, you can get turns in the animal. So you can get kind of changes in gait and things like that. So there's good reason to think that already from these systems that there's sensory encoding consistent with kind of large-scale navigation, sort of compass-like representations. There's motor evidence from cockroach suggesting that there's action-related stuff happening as well. And so really what we were trying to do was maybe connect all these dots. So what we wanted to do was we felt if you're in the middle of the brain, you have to record in behaving animals because signals tend to be basically a composite of motor and sensory variables. Plus, we wanted to eventually build up to maybe more complex behaviors, kind of like these, maybe not quite the ethological charm of this guy, who's maybe my favorite of all, but certainly, at least push the complexity of the behavior as well. And so the way we chose to do that is we developed a system where we could have the fly essentially tethered into a little platform, into a little holder, and then have a microscope objective come in, a water immersion objective come in, dissect the upper part of the cuticle a little bit, and then either come in with an electrode or just do 2-photon calcium imaging. We could do the same thing with a slightly different platform for walking on a ball. So the ball is air supported, the fly can kind of run on it, and we can use optic flow sensors to track the motion of the ball, and then that gives us the ability to close the loop. If you've seen Hannah's poster, you know that we do this in a couple of different ways. In this case, I've just shown you the LED arena, but we could equivalently do it with projectors, as Hannah's poster mentions. Okay, so what this allows us to do is open loop experiments, closed loop experiments in a behaving fly while recording. The kinds of recordings we do, so we do whole cell patch clamp recordings from identified neuron types, and then we do population level calcium imaging experiments, 2-photon calcium imaging experiments with genetically encoded calcium indicators like GCAMP. The advantage of this type of experiment is that you can record from 100% of a particular cell type, right? So you take cell type A, you're getting, it's not a statistical sampling of 0.001% or something. This is 100%. And so as you'll see, this turns out to be quite an advantage to figure out certain things. And then finally, there's the perturbing part. So, well, it shouldn't perturb you, as in this is the part about perturbing circuits. We can express proteins in there like JAWS, which allow us to shine light and then silence neurons. So optogenetic inhibitors, optogenetic activators, and so on in specific neuron types, which when we patch them, we can verify that they do silence these neurons. So this is the toolkit we wanted to throw at the problem. And then we wanted to first just see what happens, okay? So before I go to the next slide, so if there are questions, just stop me at any point, all right? If I'm going too fast, raise your hand and say that's too fast. If I'm going too slow, say you're getting bored or yawn, I'll pick up on either cue. Whatever it is, let me know, okay? All right, so the first structure we decided to focus on, primarily because it's so beguilingly beautiful, like this ring-shaped thing. So Stanley talked to you about what he calls, what they call the central body lower, CBL. In the fly, it's kind of conveniently, beautifully like a donut, so it actually closes on itself. And so that was our first target, and we wanted to see what was going on inside the donut. And we chose to focus on a very specific type of neuron. And Stanley will immediately know the type of neuron in the other terminology. CL, no, not CL. CL1, thank you. So the neurons that he calls CL1 these are conserved neurons. So we really are pretty much talking about the same neurons. So these are neurons that cut up this donut, slice up the donut, if you will, into different slices. This is a stochastic labeling experiment. And so these are labeled in different colors here. And these neurons basically send processes into one little segment of this thing. So each one has its own little arbor, okay? And so what Johannes chose to do here was actually image during closed loop walking. So the fly is walking on a ball. That's the ball over here. It's in this arena. There's a stripe in there, or there's multiple stripes. This visualization is intended to represent everything around the fly to 270 degrees behind. So it goes all the way back there. But I'm going to show it to you in a flat thing like this, just for convenience. And what you're going to see is the sensor, GCAMP6F. It's a genetically encoded calcium indicator. And you're going to see the activity in those neurons, in that entire population. Okay? So any brightening you see, is the fluorescence is the calcium indicator. So I'm just going to play the movie, and hopefully you can see what's going on. Every movement of the ball that's a turn will be reflected in the movement of this thing. Okay? So hopefully you can see what this looks like. I mean, you can imagine why this would look to us kind of like a compass, right? I mean, it looks like the activity is tethered to wherever the visual scene is in this angular sense. And we can quantitatively ask that. So we can say, if you take the activity here, can you actually predict which part of the visual scene you're in, the orientation relative to the visual surroundings? And so we did that. And so to do that, we divided up the ellipsoid body into these different slices, numbered them 1 through 16, because there really are 16 of them here. And then we took these 16 and we linearized them. So we just said, first, let's compute the activity. Let's measure the activity in each of these. Take that, get one vector that represents the population vector average of the activity in that entire torus. And then let's open it up and take the donut 1 through 16, open that up, and get something like this. So wedge 1 is here, wedge 16 is up there, okay? This is the activity profile. The brown line will be the population vector average, okay? And we're going to track that over time, and what we're going to ask is, how well does that match the angular orientation in this virtual reality world? And so what you see is it's almost a perfect match. So the correlations tend to be in the 0.9 range, or 1, or something very, very close. And so it really looks like the heading is super accurate, all is good, everything's great. Here's a question for you, though. So this is genuinely a question for the 95% of you who have not read the papers that you were assigned. So if you've read the paper and are intimately acquainted with it, please don't answer this. But for the others, so is this fair for me to conclude that this particular thing is tethered to the visual scene? Is there enough data already for you to say that? Come on. Could it be something else, or is that bump of activity really going off of the visual scene? Yeah?
Could it be due to the movement?
Right. So the obvious confound is that it could be that the fly's movements are what are driving the bump of activity, and that it doesn't have anything to do with the visual scene. So for the rest of you, and I'll come back to you if no one else does, comes up with the answer. So what kind of experiment would you like to do? There are a couple of different experiments, but suppose you wanted to compare and contrast the influence of the visual field. If you wanted to decouple things, how could you do it? What would you do? Yeah?
Do you just sort of rotate the display while not allowing it?
So you could do an open loop experiment where you just move the display and don't allow the fly to move. What else? Yeah?
There's a second stimulus.
You could do a second stimulus, sure.
You can just invert it, that when fly moves to the right, it goes to
Sure,
the
so you can change the closed loop gain and make it an inverted world. Ask me about that later, because it's kind of a cool experiment that has interesting and surprising results. So ask me about it. But we did something along those lines. So we did some of the other things that, like Andrea suggested as well, which I won't talk about. But what we did is we basically said, OK, let's take the closed loop gain. This is the normal world where if the fly is, yeah, sure.
Question from the previous slide.
Yes.
Sometimes
go back to this.
Sometimes it seems to be that that the PVA signal actually follows the panorama.
So remember that in this case, it's calcium imaging, right? So if we deconvolve it, then we still see a lag, okay? In this, so it really looks like it's sort of a post hoc reporting of what the fly should experience, not so much like, or is just experiencing, rather than a predictive thing of where it's going to go in this particular set of neurons. In the calcium imaging, as far as calcium imaging can report on that, okay? It's actually what we think is actually going on as well, but we'll come back to that near the end and you'll see why I say that, okay? Okay, so what we did is we just changed the gain. So you could take the normal world where the fly moves like this and the world moves like that, or you could take a world where, for example, the fly moves this much and the world just barely moves. So, you know, that would be a really low gain. So the blue would move barely at all, the green, you know, the fly would move a lot. Or we could make it a high gain where just a tiny movement and the whole world moves. And so your prediction would be that if it really was tethered to the visual surroundings, then kind of like in an open loop sense, it should just, the PVA, the population vector average, should track the movement of the visual stimulus. And indeed, for a large percentage of the cases, this is what we saw. So you take really low gain, and well, I should say low gain, not really low gain. You see that the brown line, which is the population vector average within this ring, which is split open again, wedge 1 through 16, tracks the visual stimulus. The angular rotation of the fly is somewhere else. Similarly, if you move it to really high gain, again, it tracks the visual stimulus and not so much the angular rotation, right? This was something that we saw consistently. The correlations are often close to 1 once again. The closed loop gain was very high. So it looks like all's well. I mean, it really is a visual representation kind of telling you stuff. Except every now and then we'd get weirdnesses where at really, really low gain, we'd notice that it wasn't holding, as in it would track the angular rotation of the fly instead of the rotation of the scene. And so Johannes did the obvious experiment, but he did it very nicely. He blackened the eyes of the fly, put the fly in pitch darkness, so no arena lights, no nothing. We checked that there's really no light in there. And then he asked, well, what happens to this bump of activity in that scenario? And so what he saw was that the compass worked just fine in darkness as well. So it really was a compass. So it wasn't just that it could represent the orientation in that visual scene and that it could track that. If it didn't have the visual scene, the bump of activity would track the fly's angular orientation. Now, I won't show you the plot on this, but it's in the paper. What you'll see is that it does it for a while, but not perfectly. So in other words, it's the equivalent of if you blindfolded me and you had me walk around. I mean, I'm capable of not smashing into something for a little while, but if I kept doing this with my eyes shut like this, eventually I'd slam into this, and that would be good for comic effect, not so good for the talk, so I won't do it. But so similarly, the fly sort of loses track over time, and without the error correction of a visual stimulus or some other stimulus, we think that the angular representation degrades. Okay, so that's what happens in this case. What was super cool was that even if the fly wasn't doing much, so this is where we get to the whole internal representation part. So already I've shown you abstraction in the sense that it can use the visual cue and it works fine. It can just use self-motion and it works fine. But impressively, from my point of view, this was kind of fun for us to see. The fly can just sit around sometimes, and then it'll sort of groom, it'll chill, and the representation just hangs around. So the next time you see a fly just sitting there, well, it could be pondering life's mysteries, right? I mean, there's a lot more going on there than, like, it's just sitting there, right? And this is not some calcium dynamics artifact. This is for 35 seconds. It can just, as long as we found the fly sitting in one place, the activity would pop back up in the right place when it resumed walking. So that's an important subtlety. So sometimes, like you saw here, the activity would persist the entire time as is visible through GCamp, which is, again, has its own sensitivity, et cetera, to deal with. But the thing that was really remarkable was that every time, even if the activity seemed to dip below our ability to sense it, when the fly started walking, almost, I'll come back to that question in just a sec, almost without fail, what you saw was points on a diagonal. So if you looked at the estimated population vector average before the fly stopped, and you compared that to what happens at restart, you get almost a diagonal. So it's as if it picks up where it left off. So it could be sitting there, the activity seems like it's dying down. As soon as it kind of perks up and walks around, in darkness, mind you, as soon as it perks up and walks around, the activity kicks back up in the right place. And so that went out to about 35 seconds. But again, this was just as long as the fly happened to hang out in one place. It's not, and then it would start walking. So yeah, question?
Do you think it will hold also after a sleep session?
This is an excellent question. I have someone in my lab right now who is quite interested in that question. It's a question that Johannes also, in his own lab, he's now at the Max Planck in Bonn at the Caesar, and he's going to explore this as well. So this is an absolutely interesting question, don't know. All right, so with any other questions? Yeah?
Did you try something a little more obvious than just increasing the gain a little or decreasing the gain initially and saw that the landmark, it wasn't following, so you tried something a little less obvious?
Less obvious, like?
Yeah, like something that was very obvious, like doing the exact opposite of what the flag
Yeah, so we'll come back to ask me that a little bit later. Yeah, that was his question. Like if you change the gain all around or you do open loop experiments, we'll come back to that at the end. I'm happy to discuss it. For now, though, the takeaway from all this was it gave us insight into what was potentially a rather cool behavioral assay that from Michael Dickinson's lab. It potentially explains what they're seeing now, which is how the fly keeps track of where it is in this little world where it's all dark. There's a central spot it needs to keep coming back, or thinks it needs to come back to. Maybe it keeps track of its orientation through this mechanism, and even if it's just standing once in a while during this little jaunt of it can still remember where it is and come back. And we think this might underlie some of these behavioral things. Okay? Any question back there, Marie? No? Okay. Sorry, confused?
Did I look confused?
Oh, no, I thought you had a question. Okay, never mind. All right, so we have a little donut here. It's tasty. We know that now. And so there's several little observations, right? So there's a single activity bump. It really does seem like an abstract representation of fly's orientation. The activity, this particular thing I should emphasize, when I say constrained to move in a ring in neural activity space, I really don't mean the physical topography of this structure, right? I mean, if you individualize these neurons and say each one's a separate unit, which it is, by the way, you can say activity always goes from this guy to this guy to this guy to this guy to this guy. They happen to be arranged in a ring. They could be spread out across half the Milky Way. As long as they're connected in this way, you'd still predict that the activity would move in a sequence, and that's kind of important. And so this kind of, I should say once more, I mean, the difference between the topology and the topography is really key. So it came up when Nakum and Eric were talking earlier about, for a map, you don't really need it to look like a map, right? It could be distributed all over and you'd still have a map. Similarly in these cells. And as I emphasized early on, and as Stanley showed, this ellipsoid body is closed in the fly, but it's not that way in other insects. In many other insects, not Diptera, it's kind of open. Finally, as I showed you, the representation persists in the absence of visual and self-motion cues. So this brings up a kind of question. So once again, for the 90% of you who have not read the paper, so I'm going to pose a couple of questions. So there's a single activity bump. How would you get a single, and Nakum already gave you the answer, so it's also a test of how close and attention you were paying to his talk. So for the 85% of you who weren't, who don't remember this, how would you get a single activity bump? You could have bumps all over the place. You have multiple visual cues on the screen. You could get bumps anywhere. If you were designing a network, what would you do to get, you'd have inhibition. So if I'm active, you shut down it. Okay, lateral inhibition, perfect. If I were to constrain it to move in a ring, you can imagine connectivity to do that too. Yes, you just connect to your neighbors maybe, okay? And if I was to say that I needed activity to persist in the absence of cues, when the animal is just standing still, what would you do then? Someone other than Paolo, come on. How would you design a network to keep activity where it is? Or give me a cellular mechanism. You don't have to give me a network mechanism. Something, come on. Plasticity? No, don't invoke plasticity. Just with activity alone.
Like a fluctuating rising action potential?
So you have an action potential and then what, riding on a plateau, you're saying? So
like open channel.
Okay, so you can have a channel that keeps it active. So you can have a biophysical mechanism. If I were to do it in a net, absolutely. If I were to do it in a network, what would I do then? Similarly. Sorry?
Recurrence?
Yeah, you have a recurrence, you have a reverberation, and then you can keep the activity up. And so the things that you guys have mentioned, basically taken together, are basically what a ring attractor is. Okay? So you have something that's an auto recurrence that allows you to keep the activity alive even if the animal is not moving or seeing anything. You have connections to your neighbors that if they're distant from you in tuning space, so again, it's important to say it's not spatial distance, it's the tuning distance. So if my heading direction is like this, if there's another neuron with heading direction diametrically opposite, I should inhibit that one, right? So it's selective inhibition of the ones that are tuned differently from you. And you maybe want to excite your neighbors so that the bump is constrained to kind of move it flows from one to the next, when the compass moves, right? And again, these can be placed anywhere. It just so happens in this case that it's placed in this structure, okay? So there's tons of, sorry, go ahead.
They are all connected together.
I haven't told you anything about connectivity yet. I'm just saying in the models, you typically assume, you don't have to, but you assume all-to-all connectivity with a particular excitation and inhibition profile, an excitatory and inhibitory profile, okay? So,
sorry.
Yes, Eric.
The thickness of those red lines implies some kind of...
Well, so in this case, yeah, so actually in this case, in the classic cosine tuning world, you'd have strong connections to the ones closer to you, and they'd weaken, and then eventually there'd be inhibition to the ones further away. And the same cosine profile can be, you can kind of move the threshold up and down so that it's all inhibition or all excitation, but it would have to be, but you need some inhibition to kind of clamp down on the activity overall. So the number of modeling studies on ring attractors and attractors in general, I mean, it's huge. This is just a small sampling. There's, a lot of it started from Chaim Sompolinski, actually in visual cortex, nothing to do with heading direction at all. He was trying to explain orientation tuning. And then it's gone from there to like there's tons of lovely models. And so the big advantage we see with the fly is, we can go from this thing, which is actually a subset of the neurons, a subset of the brain regions where people have found a head direction like cells. There's more than this. Head direction tuning is kind of all over in the rodent brain, where you have millions of cells that are interconnected in ways that we don't yet understand. And it's in principle possible to disentangle that and then find the connectivity that maybe underlies a ring attractor like network, but it's really hard. And so the big advantage we see in this particular system is we can take these models and then say, well, okay, we have strong topography. The topography is hugely helpful. The fact that I know the tuning of this cell is just shy of the tuning of this one, which is just connected, which is just next door to it and so on. That's really useful, right? Because I don't need to measure the tuning now. If I know the tuning of one cell, I know the tuning of the others just based on the location. The fact that we can monitor the entire population of a given cell type is super useful as well. You can see the whole population and actually watch this thing at work. And then if you wanted to perturb any neurons, you can go in, and if I perturb this neuron, I know I'm perturbing a neuron that relative to this one is 90 degrees off in tuning space, right? So that again is super powerful for testing models that say my tuning has to be strongly inhibitory, my connectivity has to be strongly inhibitory for a cell on the opposite side. So I can maybe potentially directly test that. And then finally, this thing I'll just kind of get to at the very end, but we have access to connectivity. So we can go in, and as I'll show you, and as Stanley actually showed you in his case as well, we can do electron microscopy, and also we can do functional connectivity experiments using sort of optogenetics and imaging to assess how different neurons are connected to each other. So this is basically what we started doing, and we in this case was Sung-Soo Kim in the lab, who's a very talented postdoc, and he worked in collaboration with Hervé Roux, who's a theorist, and Shaul Druckmann, another theorist at Janelia. And so the collaboration really worked kind of extremely well, and they went systematically together through the different criterion for a ring attractor and asked, what type of network we were actually dealing with. So the first one was mutual suppression, and so you already said, what you'd expect. You'd expect inhibition in the network, but what kind of inhibition? First, we wanted to see if there was inhibition, and so the test for that was, in this case, in a flying fly, because the flying fly also has this compass-like activity. The fly has a little stripe to fixate on at first. It doesn't always fixate in front, but moves it around. And then at some point, you'll see a bump of activity over here. At some point, Sung-Soo is going to do something which is using optogenetics, which allows him to basically selectively turn on a neuron. So he can shine red light, or in this case with a 2-photon, use a 2-photon laser at higher intensity to basically transiently excite those neurons, okay? So he's doing that while imaging at the same time. So the way he did that, he came up with a very clever way to do it. He images at very low powers. So actually, you'll see the bump a lot fuzzier here because it's super low laser powers. But just in a tiny little spot, he increases the power quite a bit, very transiently. It's just a few lines of scanning. And that alone is enough to excite these neurons in that local area. And so the question is, if you excite in this area, will you lose the earlier bump, and will the new bump take hold? Because there's inhibition in the network. Is that true? So this is what you'll see. Initial bump, red line is when it starts activated, it gets activated, and now there's a new bump in place. And the new bump actually goes on to live a happy life by itself. So it's as if you really are talking about a fly that had an initial representation of being in this room pointed in this direction, and then boom, suddenly it's, you know, the opposite, the compass is telling it it's pointed in, you know, a rather different direction. And so Sung-Soo could do this systematically and show that indeed he could basically get this effect of shutting down. So the fluorescence during the stimulation versus before the stimulation, you got an immediate drop as soon as you stimulated with the 2-photon laser. And so you saw this nice reduction of the original bump and the taking hold of the new one and it moving on. So that told us at least that there's inhibition in the network, much like you'd want. But then there's a question of, well, what kind of inhibition? What's the profile of activation here? I mean, is it local excitation? Do you just excite like things right next to you? Do you excite broadly? Do you inhibit stronger further away, or do you inhibit and maybe weaker nearer to you? What does the inhibitory profile look like? And so these are, again, this is where these kinds of details are where the different ring attractor models make different predictions. And so this is, again, an opportunity to get to implementation level details in this system. And so that's what we wanted to take advantage of. And so the first thing Sung-Soo actually did with a clever visual manipulation. So what he did was he had the fly fixating on a stripe, and then he would suddenly jump the stripe. So this is sort of like Andrea's open loop experiment, except it's within a closed loop experiment. So the fly is fixating on a stripe, the stripe is somewhere, and then suddenly, instantaneously, the stripe's going to move from there to there abruptly, not transiently, but like abruptly there. And the question is, what should you expect given the different kinds of models that we're talking about here? So these are two extreme models, right? And you basically, by jumping this stripe there, you want to ask, what happens to the bump of activity? If it's locked to the position of the stripe, if it suddenly moves, what does the bump do? Does it jump to the new position, or does it flow to the new position? Okay? That may seem like, well, okay, I mean, what does that tell you? It does tell you something, and the reason it tells you something is, if you take this cosine connectivity, and I were to suddenly move the move the bar over here, well, what would you expect?
It would just hop over.
Say it again?
Opposites.
That's great. No, the opposites is great. So you go first.
I thought it would hop over.
It would hop over, and you're saying it flows over. Okay, tell me why you think it hops over, and then we'll ask you why you think it flows.
I think it would hop over because it has connections to other cells. So the local one, it can only touch the one next to it.
So, but in this case, we're talking about the non-local one, right? Yeah. So this would be the local one, but here we're talking about something that has broader connectivity.
Yeah, so I think that's why it could have hop over because it connects farther away. Does that make sense?
But remember that the visual input in some form is coming into this network from the outside. Right? So these are the internal, this is intrinsic connectivity within the network, and now we're talking about extrinsic input, external input coming in from somewhere. So
the local connectivity model still has inhibition to all of the
sure.
cells.
Yes.
So you can jump with that model because
yes.
So the whole thing is this is like a smoother.
Yeah, okay.
So as you were saying, and you've converted now, so basically if you were to do this in this network, what you'd expect is more like a flow, right? Because in this kind of connectivity, you actually would have the bump go slowly through this whole thing, and then eventually to there. But in this kind of network, as you were suggesting, if you have very local connectivity and external input arrives there, there's no overlap between where the neurons are excited right now and the excitation that might be provided by this input. And so you'd get instantaneous sort of development of a new bump, kind of like in Sung-Soo's original experiment with the optogenetic experiment, right? It's like you excite suddenly in a different place, and you'd expect like the creation of a new bump and the diminishing of the old. So that was the prediction, right, between cosine connectivity and local connectivity. If you jump the stripe suddenly in an open loop-like thing within a closed loop experiment, you'd predict one or the other depending on what kind of connectivity there is. So now the question for you is, and now we really test who's an experimentalist and a real biologist and who's a theoretician here, and I know there's at least one from Travis's lab. So what would you expect we actually saw? Did we see, how many of you think that we saw a jump when he jumped the stripe? Wow, seriously? Or is this apathy? Okay, how many of you thought that there'd be flow? Okay, so vast majority say flow. Anyone saying jump? You're saying jump? No. Okay, one person, one brave guy says jump. That's it. Okay, wow. Okay. So I thought the biologists amongst you would sort of immediately say, of course you see both. And indeed you do. So in this particular case, you're going to see examples of both. And so he's going to suddenly jump the stripe over here, and you saw what happened. So it's actually jumping from here to there. Boom. It doesn't go through there, which you know it can go through because on the way back it does that, right? It skips that whole thing. And you'll see for contrast what happens in this case. Oh, come on. It'll sort of enlarge, almost expand into this. Okay, so it sort of fills up, it fills in. It never goes directly from there to there. It fills in. And he did this in hundreds of cases, or many tens, and eventually I think hundreds. And what he saw was, if you just used the fact that you could fit either two bumps at one Mises function, and you could fit either two things to it or one thing to it, invariably, depending on the size of the jump, you could get either, okay? But the key is the size of the jump, all right? So, yeah?
Do you see any habituation of the signal, like any kind of rundown?
That's a really good question. So we have these experiments we do where, yeah, that's a really good question. So if you look at our arena, it doesn't close all the way back, right? It closes at 270 degrees, not 360. But some people in the lab actually use even less than that. They use like 180 degree arenas. And so one interesting thing we've observed is that if the fly kind of gets used to a particular thing, so this whole bump thing, I mean, the bump takes hold very, very quickly, but how it flows through the intervening things can get smoothened with time. And so someone mentioned plasticity, and we really do think there's plasticity in this network, that repetition actually does play a role in the movement of the bump. So we think short-term stuff absolutely is important there. Yeah?
In the previous slide, there are two quantities that are changing. In one case is the angle in time. In one case is the intensity of the previous direction, right?
Sure.
Are they changing
together in predictable ways? No. So we explored pretty systematically whether we could find an easy explanation for why it would jump in one case and not others, especially from nearly the same areas, right? But there's sufficient variability in bump amplitude that you can't be 100% sure you can kind of nail that association. But what we generally saw was, and this is very similar to rodent head direction work in the sense that if you have, Jeff Taube and others have done this work, Jim Knierim, where you jump a stimulus to a completely different location and you ask, does the whole thing kind of remap to it? I mean, basically, do you get the bump moving in that scenario? And head direction cell tuning suggests that it is dependent on how far the jump is. And what we see in this case is you get jumps almost all the time if the jump is too far. If the stimulus jumps all the way there, you're going to get more often than not a bump jump to follow. If the stimulus is just moving close by, then you get something more gradual, like a flow of the bump. And so really this boils down to a couple of key variables. So there's two interacting variables. What are the two interacting variables that would determine this kind of ratio? So I talked to you about the visual stimulus as if it was a pinpoint thing coming into one particular place, but we don't actually know that, right? We don't know that a single bar stimulus comes into this network in one set of these neurons, like in a pinpoint way. That visual input could be pretty diffuse. So this input width could be anything, really. If it was wide enough, then even in this local model, you would predict that you would get flow. Does that make sense? And similarly, if the input was super sharp, well, then you could imagine jumps more often even for closer things, even if the connectivity was broader, right? So those are the two variables, and again, this is in the paper, so I recommend reading that. It's from this year, Sang-Soo and Hervé, and Hervé systematically explored this in the space of the model, in the cosine world and in this local world, the two extremes. And it was kind of cool because you could see that the model that would best explain things was actually a local model like this, because things got really unrealistic in terms of the width of the bump in the other model. So you really, in order to keep it physiological and constrained by what we knew, it really had to be sort of more local versus cosine for the kinds of input widths that we could expect, okay? So basically, we could kind of predict that this kind of model would really kind of work. Stanley, yeah?
So what is your assumption to the input width then? Because it's mediated by the ring neurons, so actually
yep.
innovate the whole ring, right?
Yep.
And so do you assume that they go to every contralateral cell?
So we are actually asking that more. Ask me that. Can you ask me that at the end?
Sure.
Okay. So, yeah.
So to study the connectivity, can't you do basic
record from two cells and see which one excite or inhibit it? Or is it possible? Or
so there's a couple of different answers to that. So firstly, paired recordings, while doable, I mean, doing them in the behaving fly is pretty tricky. Like, you know, to actually do it in the state of the network. So this network is...
You don't need behavior.
But we think you do. So we think that the assessment of connectivity you would have in a behaving animal is actually going to be different from the connectivity you'd have here. These are, I mean, as Stanley already mentioned, there's neuromodulators and neuropeptides coming in that are all, it's like imagine Eve Marder's stomatogastric ganglion moved into the middle of the fly brain. I mean, it's just flooded with neuromodulators, and we think they change the conductance levels in this circuit constantly. So assessments of connectivity, I would do, if I was looking for that level of connectivity, I would do it ideally in a behaving animal, which is hard. What we do is the poor man's version. So we do optogenetic stimulation in one place, and we measure with imaging in the other, which is kind of what you saw here. But the other thing I'll show you later, for static diagrams, we can do EM. So we can do electron microscopy level connectivity directly. So the most direct experiment for connectivity that we could come up with in this context was actually to use the same optogenetic stimulus, because as I said with this one, and as Stanley was asking, with the ring neurons that go everywhere, which, well, I haven't introduced you to the ring neurons, but just take my word for it that there are visual input neurons that go throughout the ring, individual ones that go throughout. So how local is this visual input? We don't know. So instead, what Sung-Soo decided to do was directly test this. So the direct test he did was optogenetically, so first create a bump here, and then optogenetically create another one, and have that other one either be here or here, and ask how easy is it to move the bump of activity from here to here versus here to here. If what you're saying, if the competition is such that this one strongly inhibits further away and weakly inhibits this one, it should be easier to move the bump here than there. Agreed? Does that make sense? So, okay. Well, that was what I was going to ask you. Sorry. Well, oh well, missed that chance. So anyway, so what he did was then he'd have one initial bump, and then he would compete that bump against another one either here or here, and ask whether it was easier to move the bump here or easier to move the bump there. Okay? If the network is truly inhibiting strongly those neurons that have very different preferences, this should be the harder move. What he saw instead is the amount of laser power it took to move it in either direction was almost flat. So basically, it was no different whether the bump was, original bump was 90 degrees or 180 degrees, it was kind of absolutely flat. So the conclusion for us was we're highly confident that it's actually going to be a flat connectivity profile, and that the network is essentially like this. You have local connections potentially and broad inhibition. Okay? We'll get back into this again in a little bit. All right, so at this stage, yeah, questions, Nako?
So you said you did not know the width of the input, so I was wondering if maybe you can measure it perhaps, I don't know if it's a good idea what suggesting, by creating a kind of notch stimulus, so not genetically inhibiting everything and then exciting just one segment and then introducing visual stimuli here, 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 and there, and then you might expect that, you know, depending, at some point, you know, if you modulate the calcium activity that you see in this one segment,
sure,
allowed to be active, but then as you move away,
so which part of the iceberg pops up?
Kind of, you'll be able to measure the width of this input.
Sure, that's doable. I mean, yeah, that's absolutely doable. We could do that. Again, let's come back to this question a little bit later, because it's a really cool question. The general thing of like how visual input comes into this circuit at all. Yeah, we'll come back to that. But yeah.
I just have a general question. Maybe I missed it in the paper, but if you turn off the lights and there's still the compass direction in the ring attractor, is the sharpness of the bump?
It doesn't seem to change. The bump widths are pretty much the same in darkness. When
it seemed that there was some variation in the...
No, so I mean, at least quantitatively, statistically, we can't really tell them apart in terms of, you know, broad visual scene, short visual scene. I mean, there's subtle differences, yes, but it's, you know, but it's not like this. It goes from 50 degrees to 90 degrees or something. It's kind of pretty much within a certain range, 75, 90 degrees, and so on. I'll ask David later. Yeah.
So I think in your paper you said even if you had two landmarks and they moved, to just keep that with one bump and move around?
Yeah.
So visual input's coming everywhere. How does that exactly work?
We don't know yet, but again, let's come back to the visual input thing a little bit later. I'll show you some data from the visual input thing a little bit later, but it is true that if you have two landmarks, one on this side and one opposite to it, you see interesting things. So sometimes you'd see things where transiently, right at the first time you see it, you very transiently see 2 bumps, and then one just goes and one takes hold. And that thing that takes hold will sometimes take hold, and let's say the landmark is going like this, the other one's meanwhile coming in front. At some point there's a switch. The other one's right in front, the first one's gone behind the fly, it can't even see it, and the bump is still tethered to that one. So it doesn't switch, except sometimes it switches, and it switches by 180 degrees. So we really think there is landmark tethered, visual-driven stuff happening there, but it can kind of maintain that even in the presence of direct visual input in front of it.
So I know one of the classic ring attractor properties is that if you might expect that at some level of stimulation, if you're stimulating far away, that stimulus gets ignored, right? And the same stimulation can pull the bump if it's close enough, but if it's too far away, it has.
No, so it depends on the strength of the input.
Right.
So if you give it, and that's true for us as well, so if you give it weak input, you won't even move the bump. It'll just sort of sit wherever it's sitting. But if you give it really strong, localized input, that's when we get the jumps. But it's absolutely true that if we give weaker input closer to the bump, you can kind of shift the bump much more easily, whereas in this case, it won't budge. It'll just sit where it is. Absolutely. Yeah?
If you try and confuse it a lot by giving it, using the optogenetic technique, giving it stimulus all over, does it actually show stability and go down to one?
Yeah, so in general, if you compete bumps, one tends to win. Now, in this particular situation where you're optogenetically driving those two things, I mean, it's really hard to, you can directly excite the neurons there, right? So you have to kind of ask, how strong a stimulus are you going to give? If you give it super, super strong, you can maintain them both up there. You can kind of kill the inhibition thing as well. But
does that stay after you stop exciting them?
No, one takes over immediately. So, yeah. But in general, one will win almost all the time. So, any more questions before I move on? Okay, so the next bit, how are we doing on time? So I'm an hour in, is that right? No?
Yes,
roughly, okay.
Roughly about 7.
Roughly. 7-ish, okay. So I'll take the ish part a little seriously. If you guys start looking sleepy, I mean, even sleepier than you already are, then I'll kind of stop. All right, so the next question we wanted to ask was, how does the bump move in darkness? Okay, so let's say it's a network like this. What kind of network design would give you movement in darkness? And once again, we go back to the whole ring attractor idea, right? And so one particular kind of new thing I need to introduce that's thankfully no longer new because Stanley's already introduced you to the structure. I need to introduce to you the reintroduce to you the protocerebral bridge. So I've been focused entirely on this donut, but actually tons of the magic goes on in this structure called the protocerebral bridge. And so the processes that I talked about that arborize in little parts of this, little segments of this donut, well, the other parts of this neuron, I'll ignore these ones for this talk, go up to another structure called the bridge, and there's a very particular correspondence between which column or which glomerulus of this bridge is active, is innervated, and which part of this ellipsoid body is innervated, okay? So there's a one-to-one correspondence. If this one, then that one. If this one, then that one. And it's sort of a bilateral thing. So if you go back here, this one would innervate on the other side again, and so on and so forth. So it's this interleaving pattern going left, right, left, right, left, right. So now I'm going to introduce you to another cell type, and Stanley will tell me what the correct name of this in the broader insect world is. CL2, the CL2 neurons, which we call the PEN, so this nomenclature, which actually Stanley agreed with, this EPG and PEN, or is that the nomenclature that's okay now, the EPG, PEN? Yeah, okay, all right. So the EPGs are named because of the ellipsoid body to protocerebral bridge, so that's the E-P, and the PENs are protocerebral bridge to ellipsoid body. So the reason I keep referring to Stanley whenever we talk about anatomy names is he's on this paper that's like the Insect Brain Nomenclature Committee paper. So weren't you on that paper?
No, I reviewed it.
Oh, you reviewed it, I'm sorry. He reviewed the paper that was on the nomenclature thing. So anyway, so basically these two pairs of, these two cell types, and there's actually sub-cell types within this, but we'll just keep it simple for now. The EPGs and the PENs have a particular relationship that seems quite handy for something, right? So you have these guys send their processes from here to there. If I now look at this particular glomerulus in this structure and look at the dendrites of these neurons, well, that's fine, but the axons seem to go to a different place, right? They go to the neighboring area of the ellipsoid body. If I look at this side and I go up there, the PEN neurons coming back down go to the other side. So there's this little recurrent loop, but with a shift. Can I ask for audience participation, who, again, the 95% who haven't read it, how would this potentially help you move the bump around in a compass sense? So imagine that the EPG neurons carry a bump, and now I want to move the bump around. Could you see how this would enable that? Come on. Someone. What would you need to move the bump from this place to that place?
That's a local connector.
Yep. So there's, let's say that if you went from here, let's say the bump was originally here, if you could activate this guy, he would maybe activate the side of this neuron and potentially pull the bump this side. Yeah? Does that work for you? Yes, slow nods. If I activated this guy, that would pull the bump this side. Yes?
But then it has to be limited also, because otherwise the bump is going to be flowing around this way.
If you think it might keep going that way. Okay, that could happen. So you need inhibition in the circuit as well, potentially, sure. What else would you need? So this has to happen in darkness, remember. So what you'd want is, how would the fly's angular velocity have to enter the system? What would you do there? How would you want the angular velocity to come into this? So you've just said you want to move it this way, you want to excite this guy. If you want to move it that way, you want to excite this guy. So,
I mean, Stanley's lecture, basically.
So you want, ideally, you want Stanley to lecture these neurons to tell them to. Turns out that's actually what happens practically.
So what I'm saying is like input into the protocerebral bridge from sensory.
Yeah, but what kind of input? What would that input have to, there has to be something very specific about that input, right?
Okay, so it's in darkness, so it's from movement.
Yeah, but you have to have it be asymmetric, right? The way we've constructed it, you have to, right? So that's the key thing I just wanted you to mention, which is you need an asymmetry. So it's like a seesaw. You really need one side of the bridge up at any given time, and that should pull the bump one way or the other. And if that side stays up, it should keep going, like you said, right? So you need something to stop it. And so that was indeed how we and, oh wait, I forgot to mention that. Where is that? So we were not the only ones to whom this particular idea occurred when they saw this shift. So the shift itself was noted in a beautiful anatomy paper from Tanya Wolf, who saw like there were actually 9 columns in this structure and so on. But Gabby Memmon's lab, Gabby Memon is at Rockefeller and also works in flies, and Jonathan Green and Gabby Memon noticed the same little trick here that could, the same earth-shattering or teacup-shattering conclusion that maybe this could be used to move the bump around. And so we both groups converged on a very similar model, which was essentially that if you imagine the EPGs kind of tiling this structure, and the PENs tiling the structure but with this recurrent loop with an offset kind of thing, right? So like that. If there's an initial, if you merge them, there's this offset in either direction. So if you produce a little bump of activity represented by this red blob, that should propagate up to these areas in the protocerebral bridge. If now the fly turns, let's say that one side is more active than the other, this side goes up, it pulls the bump maybe to this side. So the bump sort of shifts here, that moves the bumps up in the protocerebral bridge. If now the fly were to turn in the opposite direction, then this side would go up. And then that would yank the bump back in this way. And if it kept going, so if this activation kept up, then you'd have it keeping on moving round and round and round, okay? So this is just a conceptual model. This is not, this was just kind of an animation that we created to show how the system could work. But this made several predictions, and we kind of thought we could test those predictions. So one of the predictions is that if we record from neurons on one side or the other, what should you see? Asymmetry in the angular velocity tuning. So Stephanie Wiegner in the lab basically did that. So Stephanie patched these cells while the fly was walking in darkness and in visual in some cases, but in this case in darkness, and watched for when the cell would hyperpolarize and when the cell would depolarize when the fly turned. And so from these whole cell patch clamp recordings, what she could see is, in general, depending on the rotation, you would get either, so if it was rotating in one direction, the cells would be inhibited. These are each spikes. Each row is one trial where the velocity is indicated here. If the fly turned in the other direction, it would be excited. And so you'd get this asymmetry where it was excited in one, inhibited in the other, which you can plot as a tuning curve, like an angular velocity tuning curve. And so if she recorded from this set of neurons, the tuning went into the negative angular velocities. If she recorded from those guys, they went in the positive angular velocity direction. Okay, so you got this asymmetry there. But now I have a question for those of you who are really awake. If you believe the model, what would you think would explain what seems like large variability here? I don't mean like quality of recordings and the fact that we don't do experiments right or something. I mean, biologically, given what I've told you about the conceptual model, what would you expect the source of this variability to be? If the fly is turning one way, if these neurons is responding appropriately. I'm going to give you a little hint because this is late in the evening. I'm going to give you a hint, which is, remember that it's not just the velocity input. Our conceptual model says there's other inputs coming from the EPGs to these neurons, right? So what would you expect? Come on, yeah?
Mismatch with the path integration input?
Path integration, no. Right now, let's stick with just the stuff that we've talked about, like heading and velocity. I've kind of told you what it is. Come on. What's the variability coming from? So Stephanie's
velocity, no?
So this is the velocity that she's tracking, but what she notices is that the spike rate for what seems like the preferred velocity is sometimes low and sometimes high. It's there. I mean, it's always depolarized, but it's not always high. Why is that?
Is it moving if it's moving in the same direction as the spin or just opposite?
Nope.
No.
That's the angular velocity right there. But like I said, remember, it's not the only, velocity is not the only input here, right? Let's say there's velocity input coming from somewhere, unidentified neurons. Actually, we know which neurons, but let's say unidentified. And then let's say there's another set of inputs, which is the EPG neurons. What is cool about the EPG neurons here is that they are heading specific, right? So if my preferred heading neuron isn't giving me input, I'm not going to be firing as much, right? It's not just the velocity alone. I need a conjunction of heading and velocity tuning. So if the bump happens to be not over there, but over there, this guy is not going to get input from its EPG neurons, correct? Does that make sense? Because its EPG neurons are basically completely silent. They're inhibited, in fact. So Stephanie tested that, and so she went now to visual prep, right? So now there's a visual landmark, so there's a bump. We know the heading. We know what the heading input should be. And indeed, what she sees is if the heading and the angular velocity is on, then you get a really peaky looking response. But if it's off in a wrong heading for it, right, you get inhibition almost. The neuron basically barely fires. And so if you look across the angular velocity tuning curve, of course you get large errors here, not errors, variability here, because sometimes it's positioned in exactly its heading plus the angular velocity is right, and sometimes the fly is turned around, so even if the angular velocity input's coming in, the heading is not, right? Does that make sense? Okay.
But why does it work in the darkness?
The only reason she did this experiment in visual conditions is so that we knew the heading was known to the fly and the system. So in darkness, it works fine, as in the compass works in darkness as well. It's just we don't know how accurate the bump is in that scenario, and we're not imaging at the same time, so we couldn't infer the bump. So we wanted to do it in a visual condition.
But do you explain it with idiostatic cues? Or?
Oh, yeah. So we think that it's proprioceptive. In this case, we think it's proprioceptive input coming from the legs that tell the fly which angular velocity it has. We also think there's a role for efference, but that's a longer discussion, and we can get into that as well, meaning there are copies of the motor command being sent to the legs that also go in there. Okay, so anyway, so the conjunctive thing is why the variability looks the way it does. And so we could capture all this in a formal model, an analytical model, in this case, a firing rate model, Hervé Rowe again to the rescue. And so this model assumed nothing more than this recurrent loop with a shift, except for inhibition, which, as you've already pointed out, is essential. Otherwise, you get something that just goes round and round, or you have multiple bumps. So there was broad inhibition and this local recurrence with a shift. And so when he gives this angular velocity kind of site-specific input, what he could do is simulate a population vector average that basically was very close to what the heading would be expected to be. And the model made a particular prediction. The prediction was, well, if you look at the relationship between the bumps in the ellipsoid body, between the bump of activity that was the EPG activity and the bump of activity that would be the PEN activity, you should observe a very specific relationship between them. So here, for example, we're looking at the PENs on the right, the blue, versus the PENs on the left, that's the red. So that's the two sides of the protocerebral bridge. And one should be higher than the other if the animal is turning in one direction, right? The asymmetry that we talked about. The green represents the sum of the PENs, these two taken together. The dark blue is the EPG sum. So for now, just take for granted that one of these reds will always be higher than the, I mean, not one of these reds, the red will always be higher than the blue because we're talking about movement in this direction. But focus instead of these blue and red, focus on the green versus the dark blue. If we look at the position of the bumps, what you should see is an asymmetry where the green one sort of leads the blue in a very particular way that should become more obvious at faster angular velocities. So essentially, remember we said that it's pulled, the EPG bump is pulled down by the PENs, it's dragged around the ellipsoid body of the PENs. So we should be able to see that difference, the lag between the PENs and the EPGs in the ellipsoid body. And so what we did to test that, Dan Turner Evans in the lab basically expressed a green calcium indicator in the PEN neurons, a red one in the EPG neurons, and directly asked, do you see this lag? Do you see this difference between them when you look in darkness? And so this is the EPG neurons with the red indicator. The green one is the PENs. So you'll see here that these neurons are really only active when both the heading and the angular velocity happen. So when the fly turns is when you'll see. And play this a few times so that you get acquainted with it. But essentially ignore this for a second and just focus on the top row. And what you'll see is nothing here, and then the fly will turn, and then you'll see activity, and then will kind of keep going, it'll turn, you'll see activity some more. Meanwhile, this bump is just tracking the heading of the fly. Okay, it's going to play again, I hope. Maybe not. All right, okay, I'll play it again. So again, this one will keep track of the heading every time the fly moves, etc., etc. That one's just when the fly moves, you sort of see a bump of activity. What we were looking for was what happens down here when we kind of superimpose the two. And it's subtle because it's calcium imaging again. You should expect it to be subtle. But the green tends to lead the red around. And I'll show you the proper quantification of this. It's really subtle. So if we do quantify this carefully and we average over all these turns, what we notice is when the fly is turning in one direction, the green leads the red this way. So this is the population vector average of the green, the PEN neurons. The red is the population vector average of the EPGs. And so it tends to lead when going in this direction. And then at faster and faster velocities, it's kind of obvious. But when it's turning in the other direction, again, the green leads the red, but in the other direction. And of course, we flipped the colors of these indicators, as in we used the green indicator in one population and the red in the other to check that it wasn't a calcium indicator artifact. So this kind of told us we had pretty much everything in place, except the connectivity. We said, it's also connected. So we did functional connectivity experiments for the study itself, which I won't go into, but I'll just kind of end this piece with basically EM work we're doing with Davi Bok. So Davi Bok has basically imaged and sectioned and imaged the entire fly brain. So we have the TEM, transmission electron microscopy stack for the entire fly brain, thanks to his lab and a bunch of other people's efforts. And so within that, we can spot the ellipsoid body in this protocerebral bridge quite easily. And I started tracing, and it's very addictive because it's seldom in biology that the answer is literally right there and you just have to kind of trace it out. It's kind of cool. So you get carpal tunnel very quickly, but luckily I have tons of company, and since then more professional tracers than I have been tracing. And so this is the light microscopy image for the kinds of neurons that we've been tracing. And they include a large neuron that kind of goes everywhere. We call it the delta 7. Now this is the sectioned brain. This is one every 10 sections of the actual thing, so the skipping sections. And what you're going to see is the neurons traced out. So as you go through this, as you plunge into this volume, you'll see starting over here the neurons that we've traced. And so you can kind of go in from there. So these are the EPG neurons that arborize in the ellipsoid body, which you see there. These are the PEN neurons. And you'll see both of them kind of go up to the protocerebral bridge soon. And that's the protocerebral bridge over there. And that's the soma off to the side, as Stanley introduced you to the insect neuron. So we can essentially trace the entire thing and look at the neurons and their synaptic connections. So these are fully traced now, and all the synapses have been kind of reviewed. So we're very confident of the numbers now. Okay, this thing actually goes on and on. I'll skip the thing for anyone who wants to see a really mind-numbingly, exciting, depending on your point of view, exciting or boring movie about these neurons, just come ask me. Okay, there's a temperature warning. That's not the central complex, that's something else. The projector overheating. That movie was too much for it. No, so basically, if you look at the kinds of things we've learned from it, can we make that go away, actually, that warning? Everyone blow at it. No. Okay, for now, I'm going to continue. So one of the things we found, the first observation I'll point to is that the source of inhibition that we talked about, this global inhibition, we think there might be a very nice source for it within the set of neurons with this neuron called the delta 7, which I think Stanley talked about as the TB1 neuron. Okay, but your TB1s also get input from other places, right? Like, or no? Okay.
Well, yeah, posterior
tubercle
is not existing in flies.
Yeah. So, in this case, it's purely a protocerebral bridge kind of internal neuron. And these are neurons that Stanley's characterized in a beautiful 2007 paper in Science, where like he shows tuning. That's columns. Something's happening here. Okay, so in any case, there are these neurons, and they do actually receive the kinds of connectivity that we kind of speculated they might in that model. So they receive synapses from and make synapses to as well, which we hadn't predicted and complicates our life a little bit, from the EPGs and to the PENs. And in fact, when we measure their activity, again using two-color imaging, Dan Turner Evans again did this experiment, and Sung-Soo Kim has done it as well now. What we see is that they occupy, this is the protocerebral bridge kind of turned on its side, right and left. And what you see is that they're complementary. So when one's active, the other's down. Where the other's active, this one's down. So it's really like it inhibits everything and allows the bump to exist in only certain specific places. And so we really think that's how the system might work. But we haven't completely nailed it, but that's what we think. A second observation I'll point you to is that we did find connections between the EPGs and the PENs. So in the protocerebral bridge, which is what this represents, the EPGs do indeed talk to the PENs. And in the ellipsoid body, the PENs do talk to the EPGs, but there was a little twist. So throughout this, we've been talking about recurrent loops, like going up and coming back down with a shift. Turns out there's a little recurrent loop within this. Stanley already triggered this discussion of how there can be local processing in these little, with spikelets and so on, subthreshold responses. Well, we really do think that might be the case here, and that some part of the persistent activity that we see might be explained by very local recurrence within the ellipsoid body from back-to-back connections right here. So this is a cool little complication that we actually are kind of interested in, kind of fleshing out some more. All right, so basically this is where we're at. So I'm going to actually use more time at the end, but for this part, let me just summarize. So what we're trying to do is, we think the advantage of the fly is that you can really go to implementation level detail, exploiting basically the fact that you have this lovely structure function relationship, the fact that you have cell type specific responses, cell type specific access to the complete population of an individual cell type, and using the connectome. So an interesting question to maybe discuss later and for you to think about is, in this case, we really will have the entire connectome, and we will have probably the whole fly brain in a couple of years, maybe 5 years tops, for the entire thing at synaptic resolution. And so one thing we're already noticing from just doing a little bit in the central complex is that there are way more cell types than you need in order to do something simple like just maintain heading with this network. So what are those other cell types there for? It's horribly under-constrained. And so we really think the behavioral repertoire is underexplored, the physiological states that this network can achieve underexplored. So it's kind of pushing us in a way that we maybe didn't quite anticipate. There's loads of cell types connected in all kinds of ways that seem completely unnecessary to us, when you're looking at it from a simple point of view. But at this point, I'm going to actually continue, but first I'll just acknowledge the folks who've contributed to this part. So Johannes is kind of the guy who started almost all of the stuff I showed you. He was the first person, or the second person, to join the lab along with Eugenia, who's at Champalimo. And he and Eugenia together, were the early members of the lab, developing the initial setup to do the recordings and behaving flies. Since then, joined by many able people. So Sung-Soo Kim did the work I showed you with exploring the theoretical stuff with our collaborators, Hervé and Shaul. Stephanie did the electrophysiology. Dan did the two-color imaging I talked about. Saba is one of a few tracers. She's kind of the lead tracer for us. It's a bunch of undergraduates who kind of trace these neurons, and sometimes the PI traces them. And then Roma, I didn't talk about his work, but he's done a lot of the functional connectivity work. Tanya Wolf is the anatomist in Jerry Rubin's lab, who's kind of inspired a whole bunch of us. I don't think it's just the fly. I'd say for many people working in the central complex, her work has really been very influential. And then Hannah's work I didn't talk about because it's on the poster back there, so you can look at it. Chuntao's work I also didn't talk about yet. I may talk about in a little bit. But at this point, I mean, I'm going to stop and take some questions. So questions first.